This is the second video for the cell communication unit. In this video, we will discuss the various types of signaling and receptor molecules that can be involved in cell signaling. The binding of a signal molecule to its receptor is very similar to the interaction of an enzyme substrate and the enzyme active site. When the signaling molecule binds, it causes a conformational change in the receptor protein. It is the change in shape that leads to the activation of the receptor and the response of the cell. Most receptors exist in only two alternative states, on or off, like a light switch. Before the signaling ligand is bound to the receptor, it is off, but after binding, the light switch is turned on. Signaling molecules can be either polar or nonpolar, and receptor proteins can be found either intracellularly or on the cell surface. When signaling molecules are small and nonpolar, they are able to pass through the hydrophobic core of the plasma membrane. This means that they do not need a receptor molecule on the outside of the cell, but can instead diffuse into the cell through simple diffusion and interact with an intracellular receptor protein. Steroid signals are good examples of small, nonpolar signals whose receptors are found inside the cell. The majority of signal intracellular receptor interactions lead to changes in gene expression by entering the nucleus after binding, as shown here in the picture. Signaling molecules that are polar cannot pass through the hydrophobic interior of the plasma membrane, so they need to interact with a receptor on the exterior side of the cell. Cell surface receptors are typically more complex than intracellular receptors because they must contain a ligand binding site in the intracellular domain to interact with the signal itself. However, they must also include a transmembrane domain and a cytoplasmic domain in order to interact with both the external and internal environments of the cell. When a signaling molecule binds to the ligand binding site, the entire receptor molecule undergoes a conformational change, activating the receptor and transducing the signal from outside the cell to inside. There are thousands of different receptor proteins on the surface of any given cell, but most of them can be placed into one of three groups based on how they are activated. These are G protein coupled receptors, receptor kinases, and ion channels. G protein coupled receptors are found in virtually every eukaryotic organism. They consist of a receptor protein, which binds the signal molecule, and a G protein. G proteins are composed of the three subunits called alpha, beta, and gamma. The alpha subunit is able to bind to either GTP or GDP. When the alpha subunit is bound to GDP, as shown in the figure, then it is inactive. When the signaling ligand binds to a G protein coupled receptor, the receptor changes conformation. This active receptor is now able to bind to the G protein, and it replaces the GDP with GTP. Once bound to GTP, the alpha subunit separates from the beta and gamma subunits. The isolated GTP-bound alpha subunit is now active and able to bind to specific target proteins in the cell, activating them in turn. Those target proteins lead to cellular response that occurs in response to the signaling molecule. The body's response to adrenaline is controlled by G protein coupled receptors. The second major group of receptor proteins are called receptor kinases. A kinase is an enzyme that adds a phosphate group to another molecule in a process called phosphorylation. Receptor kinases are receptor molecules that contain cytoplasmic kinase domains. These domains become active after binding to the signaling molecule. Receptor kinases function as dimers, meaning that there must be two copies of the receptor protein for activation to occur. A signal molecule binds to the extracellular portion of two receptors, causing dimerization, or the pairing of one protein with the other. Dimerization activates the cytoplasmic kinase domains, causing them to phosphorylate each other at multiple sites on their cytoplasmic tails. These phosphorylated areas provide a place for other proteins to bind and become activated, leading to a cellular response. Insulin signals for cells to take up and use glucose by interacting with receptor kinases. The third major type of receptor proteins are ion channels. Ion channels are transport proteins which control the flow of ions across the plasma membrane. The opening of these channels occurs in different ways. Some open in response to voltage changes across a membrane, but others open when bound to a signaling molecule. These are called ligand-gated channels. When the signaling molecule binds to the channel, it causes a conformational change, opening the channel and allowing ions to travel through.